perfect. All right. Hello, everybody. Today is November 6, 2025, and this is a genetics lab. And right here, a group is working on the cure project on the cyanobacteria identification. At which step are you right now? And can I look a little bit over your shoulder if you while you do it?
That's okay. We're preparing our tubes to run them through the Qubit.
Okay, so you are measuring the DNA, specifically the DNA concentration, through a Qubit 4 fluorometer. Because I suppose you did you did nucleic acid extractions, and are now wanting to know how much DNA versus how much RNA you have, because that machine will tell you the DNA concentration specifically. All right, we'll just look over your shoulders here if you don't mind. And if at all possible, maybe you can comment what you're doing.
Okay, right now we're preparing our standard 1 tube.
Mm-hmm.
We already put working solution in it. Now we just added the standard 1.
All right, makes sense.
Yeah, that's it. So that one. So that one's ready for the Qubit. I need to...
Yeah, let's pull that waste beaker a little closer.
Oh, we have one. And then we're going to do the same thing for the standard 2.
Mm-hmm.
Okay, now we need to do our positive and our negative control. Which DNA or what Qubit DNA are we using? And we're using 5 microliters of
Mm-hmm.
DNA. This is our positive control.
So this is your sample, is that correct, or is it? Okay. Mm-hmm.
And I can store. Which one's this? Are we going to do the positive?
You want to be careful with your tip not to touch too many other things, because otherwise you'll get some contamination.
Still in the positive. And the negative we put, I'm going to just.
Mm-hmm. Yeah, that's a safer thing.
Water. The positive though, we need the DNA. I think it's like this.
Mm-hmm.
Going to use water.
Mm-hmm. Yeah, that's a good idea. That also for the Qubit fluorometry, you will use a negative control. Obviously, you will not expect to see any DNA concentration there. Yep, that's a good choice. Little recommendation, you might want to spin them down, all of them, before you, both the standards as well as the 3 samples with your little desktop centrifuge here. They will fit, yes. They should not fall through here. Yep, you have to press the button in order to open it up. Mm-hmm. Mm-hmm. You might want to just take another one just as an like a little weight. No, I think that a little amount will not make it too uneven.
I've never used this. Oh, here it is.
And that typically is sufficient.
Okay, now we're gonna run.
Yeah. Okay. Did you select the right protocol? Broad range, yes. How do you know it's broad range before you do this? Let's just check. How do you know it's broad range?
Because we don't really know, like, what the DNA is. Like, we don't know.
Yeah, well, actually, it depends on the buffers that we're using. Let me have a brief look. Do you have the broad range bottle over there, or that buffer bottle, the big one? Thank you. You see right here, it says DNA broad range buffer, BR buffer. This is why we know we want to use the broad range protocol, which is the one that you have selected. Thank you.
I'm just gonna.
Okay, standard one. Yep. Okay, standard one is in. Perfect. Now let's read standard two. Oh, I don't think you need to take a picture for the standards. I mean, feel free to do so if you choose, but the standard is not really that exciting. Then the standard two in order to get your calibration curve, right? All right, that way you have your calibration curve now, and from now onwards you can run samples. So this is the positive control. The question here is how many microliters did you add to how many of the buffer reaction mixture? And I think you had 5 to 195. If that was the case, then you just click retrieve. All right. 1.3 nanograms per microliter, but don't forget this is DNA now. This is pure DNA. It doesn't look that much, I understand. But this is DNA only, and you could go into a PCR with that amount.
Okay.
Yeah, let's compare that though. First, the negative control, good point. There we shouldn't see any concentration ideally. That machine does, of course, like there is a natural variation here. It's clearly less than the sample, and now. All right. Again, don't forget there is a natural variability both within the machine as well as these small amounts of pipetting. Feel free to run the standards again and to measure again. You'll see that you get slight variability, but the main point is your positive control and your sample are clearly higher than the negative control. So you have something in there. That's a positive sign. May I ask you, in the overall protocol, where are you right now? Are you before or after the RNase step?
We're right before. We're gonna do that next.
That's our next step.
Because to be honest, like these are relatively low concentrations as they are. To now go in with an RNase and then clean up, because whenever you put in RNase, you have to clean it up afterwards. That might remove too much DNA. In your situation, this is one of these order situations where maybe you want to try the PCR and these samples as they are. Because quite frankly, I mean, do you have the nanodrop measurements by comparison? Do you have them by any chance?
I think our other partner has them, but I can look.
Because if...
Okay, here it is from last week. From our nanodrop. I wasn't here, so I guess they were in two different tubes. We had two different samples purified, one of them.
We did it purified at 29.6. This was our final, so.
After the purification?
Yeah.
Yeah. You see, if you have 8.9 nanograms per microliter, let's just take the 8.9 nanograms per microliter on the nanodrop. And you now have about 6 nanograms per microliter on the Qubit, meaning that contain both DNA and RNA, and that is DNA only. You see that your RNA amount is not that dramatic. So you could forgo, if you choose, the RNase step and go straight into the PCR. Alternatively, what you could do now is you can say, you know what, you set up a PCR now, and with whatever remains of your sample, you go through the RNA step just to be on the safe side so that you could do an emergency PCR next time.
Okay, because we also have this much.
Is that all of our DNA?
Like, this is our other DNA. This is just the one we purified with the magnetic. But then.
And I would work. I understand. No, I would continue to work with the ones with the magnetic beads. But why don't you set up PCRs now? Run them. With any luck, they work fine, and you're good to go for the DNA sequencing. And while the PCR runs, you just do the RNAs in addition, just for backup.
Okay, cool.
Does that sound like a good deal?
Yeah.
Perfect. Thank you, ladies. I appreciate that.
Thank you.